I'd like to thank the organizers for inviting me to participate in this session. I'll be talking to you about our work trying to understand whether the peripheral immune system may be involved in mediating genetic susceptibility to Alzheimer's disease. These are my disclosures. Now, myeloid cells were implicated early after the execution of the first series of AD-GWAS approximately 10 years ago now. And in some cases, there were genes that were directly targeted by causal variants, coding variants, such as TREM2, that are relatively specific for myeloid cells. And so this cell type became of interest quite early. And this was supported also by other genes like CD33, which, while not directly affected by the variant, is the likely causal sort of gene, and is also relatively specific for myeloid cells. SPI1 is another example of this. Over time, the list of genes has expanded, and what we can see at the top left here is that if we look at the level of expression of the Alzheimer genes listed in rows over here, in either microglia in green or the cortex in general in blue, we can see that many of the AD genes are more highly expressed in purified microglia rather than the cortex itself. This is one effort that has shown this, and this was also seen in many other efforts, such as the one here where we have, instead of projection in 2 dimensions of the individual genes represented as circles, being in the expression level either in microglia on the x-axis or bulk cortex on the y-axis. In blue, what are highlighted are those genes that have a 4-fold increase in expression in microglia. And there's a significant enrichment of AD susceptibility genes within this group. So clearly from these and other studies, myeloid cells became of great interest. But myeloid cells include microglia, which of course became early a focus of studies, but also several other types of cells, including monocytes, the macrophage that differentiate from monocytes when they enter different tissues, dendritic cells, and also granulocytes. What is shown here at the top right is that when you compare different, the extent of sharing between the transcriptomes of microglia in red, one type of peritoneal macrophage in blue, or a type of stimulated macrophage here in green, we can see that there's a lot of sharing between these different myeloid cells. And that sometimes there may be unique expression to one, but there's a core sharing between all of them. And so the ambiguity remains about exactly which myeloid state may be most relevant to Alzheimer's disease. Another way to look at this is to look at the function of the genes of interest, not simply their expression pattern. One such example is shown here on the left, where the proportion of susceptibility variants that have an effect on gene expression in monocytes is shown in orange, in contrast to those that have an effect in T cells in green, and those that have an effect in both cell types in blue. And we can see that actually Alzheimer's disease is the disease that has the most polarized response in terms of innate immune responses, meaning monocytes and myeloid cells versus the T cells, which are a key feature of the adaptive immune system. So in terms of effect of the genetic variant on nearby gene expression, the myeloid cells are clearly an important target. But again, these are peripheral monocytes and they're not microglia. Similarly, if we look at different peripheral immune cells that are stimulated in a different manner, in purple here, we have different times after stimulation with different stimuli shown in individual rows. But in purple, we have memory T cells. In green, we have naive T cells. And we can see that neither of these groups of cells have any enrichment for Alzheimer's disease genes. On the other hand, the tan line rows over at the bottom are macrophages that have been stimulated with different stimuli. And so here we begin to see some nuance that, yes, myeloid cells in general are, in this case, have more open chromatin that opens up in the context of macrophage activation, but that the exact type of stimulation has an effect, and some states of stimulation are more enriched than others, particularly in this case, interferon gamma stimulation of macrophages. Further, as the number of loci that were attributable to myeloid cells began to increase, we began to have a more global picture of what may be going on. And this is nicely summarized in a cartoon at the bottom left, where we see different interacting proteins in the myeloid cell. And the ones that are highlighted in red, such as SORL1, TREM2, are, of course, the AD susceptibility genes. And so what we're beginning to see is that these different variants that anchor different susceptibility loci and the genes that they affect may all be affecting a related cellular function potentially in myeloid cells. This has been further delineated by the fact that if you perturb one AD susceptibility gene, such as SPI1, PU.1, you actually affect other AD susceptibility genes in the myeloid cells like C33, some of the MS4A genes, ApoE, and clusterin. This suggests again that there are interconnecting functional relationships between these different loci in the same cell type. Similarly, more recently, the MS4A locus have been shown to influence the expression level of soluble TREM2 in the spinal fluid, again showing how different loci affecting the same cell type may be working together in different ways. So what is the evidence today that that points us towards primarily microglia versus monocytes, or perhaps a combination of the two in the onset of AD in terms of the effects of the susceptibility variants? So we set about to evaluate this question by interrogating the peripheral immune system in cohorts of well-characterized individuals. So for this, we used samples from two cohorts, the Religious Order Study and the Memory and Aging Project, both studies developed and run by Dr. David Bennett, and for which all the neuropathological characterization has been done by Dr. Julie Schneider and her team. Both cohorts were started in the 1990s. The subjects are free of enrollment, for the dementia at the time of enrollment, and they agreed to an anatomical gift act, so donating their spinal cord and brain at the time of autopsy. Now, the samples of blood were cryopreserved over the years before people passed away. And so we had the opportunity to take these peripheral blood mononuclear cells and to sort out the monocytes from them. We then miniaturized cDNA library construction so that we only needed 2,000 monocytes, and generated these data in 238 subjects from Ross and MAP, generating about 25 million paired end reads per subject. Importantly, all of these subjects had neuropathological data from the structured neuropathological examination that is performed at Rush, and also had RNA sequence data from the dorsolateral prefrontal cortex, which we had generated in earlier studies. In addition, another subset of subjects had an interesting phenotype, which is the proportion of activated microglia. This is a score that is determined using the morphology of individual microglia, with the activated ones having a stage 3 or a more globular, less ramified appearance. And 59 subjects had that particular trait. So we first asked the question of whether the monocyte RNA sequence data was a useful proxy for cortical RNA-seq data. And the results are shown here on a per gene basis. And the basic answer is that there essentially is not a good correlation or any meaningful correlation between the two sets of RNA-seq data. There is a subset of genes shown here in orange where there is a significant correlation between monocyte expression and cortical expression. However, when we looked a little bit more closely at this subset of genes that have a correlation between the two tissues, we find that they are actually the target of very strong cis-QTLs, and this primarily explains the correlation. So here, shown here again, is the effect of the sorry. What's shown here is the effect of the genetic variant on a cis expression in monocytes on the left, frontal cortex on the right, with the p-value of the correlation being shown in the y-axis, and on the x-axis is the extent of cross-tissue correlation. Again, we can see that most of the correlation here is really driven by genetic variability, genetic variants. So again, there's a little correlation at the RNA level between the two tissues, but perhaps the monocytes could be useful as a biomarker to capture something about the Alzheimer's-related traits in the brain. And so we did first a transcriptome-wide association study for Alzheimer's disease of endophenotypes, including what's shown here is amyloid pathology burden. We can show here is the entire genome-wide set of results on the x-axis. The y-axis is the level of significance, with the orange line denoting the threshold of statistical significance. And very clearly, there are no single genes that are associated with the burden of amyloid pathology in monocytes. The same is true for tau pathology and for our measures of the slope of cognitive decline prior to death, and also a syndromic diagnoses of either pathological AD or AD dementia. So again, very little evidence that the transcriptome of the monocytes in the periphery are relevant to some of these Alzheimer-related traits. However, there are associations to other traits, most prominently to intracranial atherosclerosis, which is shown here. And here again, you see a large number of genes, over 100, that are associated with the extent of atherosclerosis in the intracranial circulation. This makes sense since monocytes and macrophage are well known to participate in the accumulation of atherosclerotic plaque. Now, of course, the single gene analysis is limited by the problem of multiple hypothesis correction and the high statistical burden that we need to meet. So we also use WCGNA to identify modules of co-expressed genes to reduce the dimensionality of the data and to perform the same types of analyses. So out of the 9,000 genes that we had measured in each individual, we identified 20 modules of co-expressed genes, which are shown here, with each color representing one module and each rectangle representing one gene. We also show that some of the modules are similar to one another, have some correlation with one another, and this is shown here, while others, like the brown module, is not particularly correlated with any other module. When we look at the associations between a meta feature of each of the modules and their outcomes of interest, we again find no meaningful association with the traits that are directly relevant to Alzheimer's disease. We do find an association of the pink module, which is shown here at the bottom, with diffuse amyloid plaques. So this again is a very early phenotype. It is different from the neuritic amyloid plaques, which also has tau and are more closely related to cognitive decline. And interestingly, we find a different module, the black module, as being associated with Lewy bodies. And again, not surprising, we see a large module consisting of genes that are related to cerebral atherosclerosis. This picks up the associations that we saw earlier. We also looked at the trait that I mentioned, the proportion of activated microglia. And what is shown here is that multiple different modules are seen to be associated with this particular trait. So this suggests that there is some form of communication between the peripheral immune system and the central immune system, in this case, the activated microglia. And we can see that in general, these modules are all interconnected with one another. So in summary for this part of the talk, we find that monocyte gene RNA expression is generally poorly correlated with postmortem brain RNA expression. So peripheral monocyte RNA expression is not a proxy for brain expression, even for genes that are relatively specific for microglia. Monocyte gene RNA expression is most strongly related to cortical microglial activation and cerebral atherosclerosis, but not the Alzheimer traits. However, both of these traits are relevant to Alzheimer's disease, as vascular disease modifies and is an important risk factor for AD dementia. And of course, microglial activation has previously been shown to be associated with AD. So some of the lack of AD associations may be due to sample size, and we'll need to scale up our studies to see whether more modest effects may not be existing. But overall, I would say this data suggests that the monocytes may be less likely to be involved in the effects of genetic variants than microglia that are resident in the brain. Nonetheless, monocytes have another function, which is to serve as a model system that is much more easily manipulable than the microglia in the central nervous system, which are difficult to access. And so, as one example, what we use this this similarity between monocytes and microglia to look at how the different genetic variants associated with AD could affect the expression at the protein level of some of the AD genes. And so we measured several of the AD genes, including C33, TREM2, PTK2B, through cytometry using fluorescence-labeled antibodies. And we did this in healthy subjects, since again, we were interested in the context of people before they had the disease. So we looked at both young individuals from the INVAR project and older individuals from the Harvard Aging Brain Study. This initial project was published, and so this is simply the summary of what we found, that there was, again, the cis effect of the C33 variant on protein level expression of C33, which we had described previously. But very interestingly, we found that C33 actually also influences TREM2. And we were able to validate this by showing that if we perturb the protein level expression of C33 with an anti-C33 antibody, this affected the level expression of TREM2. Similarly, we found that the NME8 locus had an effect on PTK2B expression at the protein level. Interestingly, in both cases, there was no relationship at the RNA level. This was only occurring at the protein level and highlights the need for more protein QTL type studies. More recently, we reanalyzed these data, this time at the genome-wide level, to discover novel loci that may be influencing the expression of these very interesting proteins. So we rediscovered the C33 and TREM1 loci as being affecting the expression of C33 and TREM1. So these are cis protein effects. They're basically positive controls. But surprisingly, we found that the same locus, the SERP locus, and more specifically the SERPB1 gene within that locus, was influencing the expression of two different Alzheimer genes, TYROBP and TREM2. And interestingly, of course, these two proteins rather, interact with one another and are a known ligand receptor pair. This is GWAS based on a relatively modest number of subjects, and shows the power of leveraging monocytes as a model system. Given our short amount of time, I'm going to skip ahead and just to show that when we perturbed the level of expression of SERPB1 to try to validate the genetic association that we had found, we also reduced level expression of TREM2 and TYROBP. We also affect the level of uptake in the myeloid cell. And so here we have, again, the little diagram to summarize the results, where one locus, which is not known to be associated with Alzheimer's disease, although SERPB1 does harbor a variant that was suggestive, a rare variant that was suggestive for being involved in AD, but has not yet been validated. And this affects both SERPB1, which then it goes on to influence TYROBP, and separately affects TREM2. The mechanism is not yet completely worked up, but again, we have now connected these different AD genes with one regulatory locus elsewhere in the genome, a locus and a gene that has been previously implicated in microglial phagocytosis. And so again, this makes sense intuitively, since we are affecting one of the key features of myeloid function, which is probably involved in regulating amyloid biology in the aging brain. With this, I'll summarize. And so again, overall, our monocyte study at the RNA-seq level revealed that there is certainly no strong connection between to the transcriptome of monocytes and endophenotypes related to AD, either in the brain or with cognition. However, these associations are relevant to cerebral atherosclerosis, the level of activation of microglia, and perhaps some other neuropathologies of aging, including Lewy bodies and diffuse amyloid deposition, which will have to be further dissected. So today, are monocytes or microglia more important in terms of the effect of AD susceptibility variants? I would say the jury is still out, but current evidence clearly favors resident microglia as the primary target of the AD variants. But again, we'll have to do further analyses to demonstrate this more completely. Nonetheless, primary monocytes, because they share so much of the transcriptional machinery and the other functions of microglia, are an excellent model system, especially to do human genetic analyses with which to dissect the relationships between different AD susceptibility variants, as I showed you in the case of SIRPV1. With this, I'd like to thank you for your attention and to recognize the key work of some key members of the lab, including Dan Felsky and Yi Ma, who have been working on the monocyte data. Dan now has his own laboratory in Toronto. And in terms of the work on the monocyte SIRPV1 story, this was work done by Charles White, while we were in Boston. Thank you very much, and I'll take questions.